Hi, hello. Very pleasure to see all of you in this video lecture. In this video lecture, we will discuss about phylum Platyhelminthes, that is usually termed to be flatworm. Now we will see the general characters. After that, we will see the different classes of the phylum, and then we will discuss about tapeworm, Taeniasolium. And also two different type of tapeworm is that, Taeniasolium and Taenia saginata. The difference between the two will be asked predominantly in the Neet questions which are already asked. And then we will see about tapeworm life cycle as well as the life cycle of Fasciola hepatica, liver fluke. And then some important concept of planaria, regeneration capacity. These are the three very important areas where the question setters are looking into it. So be aware of these three important subtopics. Let's discuss with the general characters. These Platyhelminthes are mostly endoparasites. That is tapeworm, which is a flatworm. They are all parasites. They found in animals including human beings. Yes, as you may know that Taeniasolium, which is present in the intestine of human. So they are all the parasites. And flatworms are bilaterally symmetrical animals. That means next level of development from porifera. If you see porifera, coelenterata, ctenophora, they are all radially symmetrical. They are from lower group of animals, where here there is a difference. There is a movement from lower to higher group. So bilaterally symmetrical animals they are, and more than that they are triploblastic. That is ectoderm, endoderm, mesoderm, 3 germ layers are there. Here there is no coelom, acoelomate organism. This is acoelomate animals. Okay, and the next level is flatworms are organ level of organization. Porifera is cellular level of organization, and then coelentrates are tissue level of organization. Now the flatworm are organ level of organization. There is a gradual development from the lower group to the higher group. Here the sexes are also not separate, hermaphrodite, that is male and female organ present in the same individual. ஒரே ஆர்கனிஸ்ல ரெண்டுமே இருக்கு. Fertilization is internal and development through larval stages. Internal means within the body fertilization will take place. And here very important property is flame cells. Flame cell play a very important role over here. These flame cells help in excretion. The body waste will be excreted through flame cells. This is a very important property of Platyhelminthes or flatworms. Hooks and suckers are present in the parasitic forms. I will show you the hooks and suckers in the organism. In the head portion, and that is made up of scolax that bear suckers and hooks. Suckers will help the parasite to get the food into it. Hooks will help the organism to cling into the host organism. That is the role of suckers and hooks. And moreover, planaria which possess high regeneration capacity. That means if we divide the planaria into many different parts, each part can develop into the adult planaria. That is the regeneration capacity which we already saw in the hydra, but the power of regeneration capacity is more in case of planaria. You just keep in your mind. Now we will discuss with the three important classes of Platyhelminthes. One is Turbularia, next one is Trematoda, third one is Cestoda. Turbularia is a fish and also marine water organism. The regeneration is common in this case. As we saw planaria, this is this name, planaria scientific name is Dugisia. They also belong to the class Turbularia. So regeneration capacity is common among Turbularia group class. Trematoda, this is an parasite. Either it can be a endo or ectoparasite. Flukes. Body bears one or more suckers. Suckers will help to get the food from the host. Here there is an example, Fasciola hepatica, liver fluke. This is one is liver fluke. And next one is Schistostoma, that is blood fluke of man. Fasciola hepatica is liver fluke and Schistostoma is blood fluke. You just know the other names also because that can be asked in the Neet. Cestoda, they are also a parasite. This is a endoparasite living within the digestive system, especially intestine. As we already discussed, Taeniasolium that belong to this group, Cestoda. Hymenoplepsis nana, that is the dwarf tapeworm. This all belongs to the class Cestoda. Here the body is generally divided into few or many proglottides, not true segments. Let's we discuss with the structure of the animal. That will help you more things in order to understand what is proglottides, what is sucker, what is hooks, like that. Here this is an Taeniasolium. This is the head portion, and the primary host of Taeniasolium is usually pig or cattle, and it is more prevalent in beef eating persons, Taenia group. It has neither mouth or anus. It absorbs food throughout the body surface. Here this Taenia is divisible into three parts. Like head, here scolex is there. This scolex bears 4 suckers. Suckers help to imbibe the food materials from the host, whereas hooks helps to cling the host, which helps them to take the food from the host. So there are 28 to 32 hooks and 4 suckers are there. Small apical part of the scolex is termed to be the rostellum. There are 2 important groups, Taeniasolium, Taenia saginata, 2 individuals which belong to the group of Taenia genus. One of them, Taenia saginata, does not possess hooks on its scolex. They does not possess hooks. Here you can see this is Taeniasolium, and this one is Taenia saginata. Here Taeniasolium possesses the 4 suckers, 1, 2, 3 and 4. At the end of these things you can see there are many hooks. This region is termed to be rostellum with 28 to 32 hooks will be there. Whereas here there will be no presence of rostellum, only 4 suckers will be there, no hooks, rostellum. This is the very important unique feature of unique difference between the Taeniasolium and saginata, which are asked in the Neet questions. Here the neck is unsegmented, they are budding, it is the budding zone. And strobila, this is the this is the body and the neck portion, and this is head portion, scolex. This strobila body is made up of many segments. Like this is not two segments. If you say true segments that belong to Annelida, that is termed to be the metamerically segmented body. Whereas here, this is the proglottides are not true segments; they are pseudo segment. So they are said to be pseudo metamerism. Pseudo means false. Here there are two portions, anterior and posterior and middle portion. The anterior proglottides are immature and middle proglottides are mature, whereas posterior one are ripe or gravid. These ripe or gravid proglottides, which are present in the posterior region, regularly detach from the strobila, and also they pass out in the feces of the host. This process is known as apolysis. Here the very important feature is self-fertilization. As I already told, both male and female organ of the animal present in the same individual. So self-fertilization, that is the egg of the male female organ and sperm of the male organ, both will fertilize in the same organism. This is very unique feature. So self-fertilization here it is possible. If the tapeworm enters into your body, they may not enter with a pair. It may remain as a single, but it will lead to many number of tapeworms in your intestine. Am I right? How this is happening? This is because the tapeworm possesses the ability to fertilize self, that is self-fertilization. Male and female of the same organism will produce the respective gametes, sperm and egg, and they fertilize, result in the zygote that will give rise to the next tapeworm without the help of any pairs. Now here this is a tapeworm with head that is scolex, and the body surface is there. The scolex was elaborately described here, that the scolex possesses suckers, like that 4 suckers will be there, and the head is this is rosatellum, and hooks are there. You can see the hooks in the head. Now this is the difference. Taenia solium possesses rosatellum and hooks, whereas Taenia saginata does not possess rosatellum and hooks. Here see the another important difference between Taenia solium and Taenia saginata. In Taenia solium, 12 uterine branches, major uterine branches will be there, whereas in Taenia saginata more than 13 uterine branches will be there. Here see. So this is the main important difference. Here more than 13 uterine branches, here only 12 branches. These two important feature is the difference. One is uterine branches and the next one is rosatellum hooks. Taenia solium possesses the rosatellum hooks, whereas saginata does not possess. Here uterine branches are in Taenia solium it is 12, whereas in saginata it was more than 13. In Neet question papers, they used to ask what are all the larval stages present in the life cycle of Taenia solium or other groups present in the flatworms. So you must be aware about the life cycle of Taenia, tapeworms. Taenia means tapeworms. Taenia solium means that is one member, one individual, and Taenia saginata is another member. Here let's we see about the tapeworms. There are three important larval stages. One is zygotes will develop into the oncospheres. This is one type of larva, and these while during elimination of the waste, these oncospheres from the host will be released out, and this will be eaten by the pigs. This oncosphere contains hexaconth, which is the special region present within the oncosphere that can able to penetrate into the gut, blood and also muscle of the pig. This is the special important thing, oncosphere hexaconth. This hexaconth will develop into the cysticerci larva. This cysticerci larva will enter into our human body, and it will reach the gut region or portion of the humans. Here. So how many larvae are there? Oncosphere, hexaconth and then cysticerci larva. These three important larval stages are present in the tapeworm. Now we will see the life cycle over here. Humans infected by injecting raw or uncooked beef or pork. Here Taenia saginata will always present in the beef. So uncooked beef will result in transfer of saginata towards our human intestine. Similarly, other foods, solium will be there, cattle. In cattle or beef, tapeworms will be there. By this process, human intestine will get infected by this tapeworm. Now see the feces from the humans have eggs or gravid proglottides in the feces. These gravid proglottides or eggs that can be developed into the oncosphere, that is we have already saw oncosphere larva that can able to inject that can be able to infect the cattle and also pigs. In case of Taenia saginata, that will affect the cattle, that is beef, like that. In case of Taenia solium, that will infect the pigs. But the type of larva is same, that is oncosphere larva. Here, these oncosphere hatch and penetrate intestinal wall and circulate in the muscle. Oncosphere possess hexaconth that will can able to do all this process. This oncosphere will develop into another larval stage called cysticerci larva in the muscle. This larva, cysticerci larva, will enter into the human. So the question can be asked from this portion: oncosphere, hexaconth, cysticerci larva. Three larval stages present in the tapeworm. Here in tapeworm, the primary host is either cattle for Taenia saginata, that is beef, and the next one is pig. And the intermediate or secondary host is humans. Now, what are all the disease caused by tapeworm in human? One is Taeniasis and the next one is cysticercosis. The cysticercosis is more dangerous than Taeniasis, which is caused when a person incidentally ingests the egg or oncospheres with contaminated food or contaminated water, or even through your fingernails. This hexaconth usually will reach the eye portion or brain to develop into cysticerci, that is known as cysticercosis, where here this cysticerci will cause the blindness and also the brain necrosis thing. So cysticercosis is more dangerous than Taeniasis. This Taeniasis will be present in the digestive system, whereas cystic cirrhosis will reach the eye and the brain and cause blindness and brain necrosis. So be aware of this. And the next one is fasciola hepatica, that is liver fluke. Here in this session also they can ask the question. You just know what are all the primary host and secondary host. Here the primary host will be sheep, goat, cattle, pigs, deer, rabbits also. Here the intermediate host is snail. You must aware of what are all the larval stages of fasciola hepatica and which type of larvae present in the primary and secondary host. Here this is an cattle which is eating the grass, and by means of grass, in that grass metacercariae, that is the larval stage of fasciola hepatica, eaten by the animal, and it is present in their body. This will be excreted in the feces of the particular cattle. Here in the feces there will be a larval stage. That larval stage is termed to be miracidium. This miracidium will move and attack the mud snail which is present in nearby. In that snail, it will reach the sporocyst form and then rediae and cercariae form by means of parthenogenesis. That means the spore will develop into another form without any fertilization between male and female, sperm and egg. So that is termed to be parthenogenesis, sporocyst, and then rediae and cercariae, these three forms, SRC. These three forms are very important. These three forms are developing inside the snail itself. Snail is the secondary or intermediate host. Okay, this miracidium becomes SRC in the mud snail. After this, it will become attain the larval stage, another larval stage, that is termed to be cercaria. After 5 to 7 weeks, it will become cercaria. This cercaria will reach the grass. This cercaria will reach the grass and then develop into the next larval stage called metacercaria. So here the larval stages are metacercaria, and then from the feces of animal, miracidium, and in the secondary host, sporocyst, rediae, cercariae. These cercariae in turn develop into the metacercariae. Likewise, the life cycle will go on for fasciola hepatica. Then we will see about the questions which are already asked in this portion. The excretory structure of flatworm or Taeniae are excretory structure, which is the famous one, flame cells. Taeniae saginata differs from Taeniae solium in, I already told, the rosatellum hooks will not be present in the saginata, whereas in Taeniae solium it is there. Here hooks present in Taeniae solium, not in saginata. So what they are asking, absence of scolex hooks. Scolex means head. So this is right, but in addition uterine branching is seen in the next option. We have discussed 12 uterine branches in Taeniae solium, more than 13 in saginata. So there will be a difference in the number of uterine branches among solium and saginata. So option 2 will be the perfect right answer for the question. This was asked in Neet 1990. Which is correct about Taeniae? In 92 they are asking. Which is correct about Taeniae solium? Male organs occur in the posterior proglottides. male organs occur in anterior proglottids or female organs occur in anterior proglottids. Mature proglottids contain both male and female. The fourth option is correct. And the next question is, in contrast to annelids, the platyhelminthes show. Here what is the difference in contrast to annelids? There is absence of body cavity? No. There is presence of pseudocele by radial symmetry or bilateral symmetry. Both of them are bilateral symmetry, so the answer will not be in the symmetricity because both of them exhibiting bilateral symmetry. So the answer will be in absence of body cavity in Platyhelminthes. And the next question, which one of the following kind of animals are triploblastic? Sponges, diploblastic, Ctenophora, diploblastic, coelentrates, coral sea anemone or coelentrates, they are also diploblastic. The only one is which is triploblastic is flatworm or Platyhelminthes. Transfer of Taeniae into the secondary host occur as, in which form Taeniae will transfer into the secondary host? That can be asked. I already told. Transfer of Taeniae into the secondary host. It happens as the oncosphere. Through oncosphere, the Taeniae will transfer towards the secondary host. I hope this session will give you benefit for you. If you like this video, don't forget to subscribe the below button and also click the bell symbol. This will help you to remember whenever I host new videos, it will come into your cabin. Thank you. See you later in the next video.